My name is Stefan Steiner. I'm the chair of the Statistics and Actuarial Science Department here at the University of Waterloo and the host for this talk. So before I move to the talk, I just want to give you a little bit of background and about this seminar series. It's the David Sprott Distinguished Speaker Series. And not too surprisingly, this honors David Sprott, who in 1967 was the founding chair of our department, and as it happens, also the founding dean of the Faculty of Mathematics here at Waterloo. Throughout his long career, he played a fundamental role in the success and growth of the department, which is now the largest in Canada and one of the largest in the world. The goal of the seminar series is to invite outstanding researchers in statistical sciences to deliver a lecture. Normally, this also includes a visit to Waterloo, but for obvious reasons, this isn't possible this year. Just wanted a couple items for logistics. One is that we are going to be recording this session, so that's already started, just so you're aware. I'll now move to a brief introduction to Trevor, who really probably doesn't need much introduction. Most of you, I'm sure, are familiar with Trevor. So he's on faculty at Stanford University, and he's been there since 1994, and is currently the John A. Overdeck Professor. He holds a joint appointment in the Department of Statistics and the Division of Biostatistics of the Health Research and Policy Department in the Stanford School of Medicine. And before that, he was a member of the technical staff at AT&T Bell Laboratories for 9 years. Trevor's main research interests or contributions have been in applied statistical modeling of predictive problems in biology, genomics, medicine, and industry. And I'm sure you're aware, but he's been incredibly productive. He's published over 200 articles, 6 books, and according to Google Scholar, his work has generated more than 250,000 citations. His books are, of course, very well known, and many of them are classics, and I'm sure you've read many of them or looked at many of them. Books such as Generalized Additive Models, Elements of Statistical Learning, and more recently, Computer Age Statistical Inference, which brings me to a more personal point, is that I was really impressed with his most recent book. And here at Waterloo, I helped organize a reading circle to go through the book in more detail, which was really fun. So one more thing before we move to how we're going to handle questions and so on is that if you have a question that you'd like to speak orally, then you could please raise your hand in the chat, or you could just type in your question in the chat, and we'll try and deliver that question to Trevor at the appropriate moment. In any case, without further ado, I want to really thank Trevor Hastie for agreeing to give this talk, and now turn it over to him for his talk on predictive models in health research.
Good afternoon, everyone. Thanks for the nice introduction, Stefan. And also thank you to the department and to Greg Preston and Chang Wu for organizing this session. And thank you to the department for selecting me to be the David Sprott Distinguished Lecturer for 2020. It's a real honor. I spent some time going through the list, and I saw some of my heroes there. So I'm really pleased to be included amongst them. And even though I'm not the first David Sprott lecturer, I think I may be the first one to give the lecture in my sheepskin slippers. So I'm going to share my screen, and let's see if we get that to work. Yeah. I'm sharing preview, and now I'll just get it to full screen. Is that good, Stefan?
Looks good, thank you.
Okay, good. So this talk is about predictive models in health research, and it's especially about using some of the modern tools, a lot of them developed in machine learning, in the context of health research and biomedicine. So I'm going to cover a few topics, but mostly this talk centered on supervised learning. So building models from data that predict an outcome using a collection of input features. So this is a topic I've been working in for a long time, and some of our books cover these areas. So there's some powerful and exciting tools for making prediction from data, right? And especially these last years, it's been a very exciting period. But these tools aren't magic, and so one should be skeptical. They require good data and proper internal validation. They're very powerful, right? And so you can pretty much get any results you want, but if you do proper validation, you'll tend to be more cautious. Human judgment and ingenuity are essential for their success, and they may be overkill. If simpler and more traditional methods do as well, they're preferable, and it's known as Occam's razor principle. So that's a message. Some definitions: machine learning, constructs algorithms that can learn from data. Statistical learning is a branch of applied statistics that emerged in response to machine learning, emphasizing statistical models and assessment of uncertainty. And then more recently, data science is the extraction of knowledge from data using ideas from mathematics, statistics, machine learning, computer science, and engineering. But really, all these are very similar with different emphases. And, from my point of view, this is really just applied statistics that's moved into the 21st century. So another take-home message is to stress the importance of internal model validation. So, and this is really important. So don't trust me or anyone who says they have a wonderful machine learning algorithm unless you see the results of careful internal validation. So for example, you divide the data into two parts, A and B. You run the algorithm on part A and then test it on part B. And obviously the algorithm must not have seen any of the data in part B. If it works in part B, you have some confidence in it. Okay, so this seems like a very simple idea. Is it done properly in practice? Rarely, unfortunately. So in God we trust, all others must bring data. This is a statistical proverb, sometimes attributed to W. Edwards Deming, but others have precedent, and it goes back to Edwin Fisher, 1978, and even before him. But now, really need to stress this point. It's so easy to get excited with some of these modern tools, and you feel like you can get such strong results on your data. But if you do a really honest validation, that's going to sort of that's going to make you behave in a little bit more responsible way and make sure that the results are real. Okay, so modern data vary in shape, and these call for different approaches. So here's some cartoon images of data shapes. So we got wide data. So this we're thinking of this as the feature matrix, right? So the rows are observations and the columns are variables, right? So with wide data, you've got thousands or millions of variables and hundreds of samples, right? So we see data like this. This came up first, say, in genomics, in image modeling, nowadays in web modeling. And you got more variables and observations. You need to do some kind of regularization or screening. False discovery rate, that's like univariate screening in genomics, is used a lot. Lasso, which does selection of variables, or other kinds of regularizations. Support vector machines manage well, surprisingly, in data of this kind, and maybe stepwise methods for doing variable selection. And we contrast that with tall data, where you may have tens or hundreds of variables, but you've got thousands or millions of samples. Data sets like this come up often in business applications, also in web applications. And, and so here we can afford to fit more adventurous models, right? We can fit interactions, we can fit nonlinear models. And so there's tools that have been developed that really take advantage of having lots of data, such as random forests, boosting deep learning, and even generalized linear models, but with a lot of extra structure built in through transformations and variable expansions and so on. And so these two, these two types of data have been around for the last 20, 30 years, and we've had to learn ways to deal with them. But now, we also get tall and wide data, right? So thousands or millions of variables, millions to billions of samples. So these are truly massive data sets. Certainly can't fit them in memory, but you need to find ways to deal with them. And I've encountered data sets like this, and many others have as well, the so-called big data, what you do with it. And there's a lot of tricks of the trade. So ways of exploiting sparsity, often big data sets like this are very sparse. Random projections or hashing, variable screening, subsample rows, divide and recombine, case control sampling. There's just a whole bunch of techniques that have evolved to take care of data like this. In deep learning, stochastic gradient descent is the common way of fitting, which also does subsampling of the rows. So for the remainder of the talk, I'm going to focus on 3 types of examples, and they're going to visit each of these types of data. So the first example is large-scale Lasso models using the package GlimNet, which we've developed here in Stanford for fitting models of this kind. And we'll work with the examples that I'll show you are both tall and wide, massive data set. Random forests used as an ensemble learner, that's for tall data, and deep learning used for image classification, which is wide data. And then I'll give an example of machine learning overkill and end the talk. Okay, so GlimNet is a package we've developed. It's been, I think, going on for over 10 years now at Stanford. This fits what it does is it's an R package, and it fits a regularization path for a variety of generalized linear models with Lasso and elastic net penalties. Okay. And I'm personally very proud of this package, and I put a lot of effort into it, and it keeps on getting better. And you can see the team of people that's involved. This is meant to be me, the supervisor, and we got Jerry Friedman, who wrote a lot of the initial low-level code in Fortran, actually, for GlimNet. Rob Tibshirani, we were the initial members of the team. There's Noah Simon, and these were graduate students. Noah Simon, Yunyang Kian is one of my PhD students. This is Balasubramaniam Narasimhan, who's a researcher in our department, and Ken Tay, who's a current graduate student who's made very, very nice innovations in this package with me. And I want to stress, because this project is largely to do with computation and not so much statistics research, but it's really important, for statisticians, computational tools are really critical to doing our job, right? And having good tools, good packages is really important. So time spent developing packages is a great service to the community, and honestly, it deserves some kind of recognition. So for example, Ken Tay, this student on the right here, came to me about 6 months ago, and he said to me, and he's a PhD student, so he's required to write papers and so on. He said to me he really wants to spend, he wants to volunteer some time to help expand this GlimNet package. And so I said, great, let's do it. And so he's worked with me, and he's putting a lot of time. I'll tell you some more about the things that he's helped develop to augment this package. And I think, it's really, you probably won't get a publication out of it, but you'll get a lot of recognition on the package website and so on. So that's just a little plug for spending some time on computational research. So here's a logistic regression model, which is a common GLM that we like to fit. And, if you've got wide data, lots of coefficients, the Lasso penalty, which I'm sure we all know about, developed by some Canadian guy in 1996, induces sparsity in the coefficients through an L1 penalty. And it shrinks them towards zero and sets many of the coefficients to zero. And it's an automatic way of doing regularization and variable selection. And so that's sort of the driving model underneath, well, the penalties, it drives the whole package, and along with the elastic net. What this package does fits a regularization path, so you'll have a grid of values of S, usually 100, and that covers the entire regularization path from when everything's zero to the unregularized fit. It uses, and it fits this path efficiently using coordinate descent, which is a very old method, but works extraordinarily well in the context of this penalty, because it operates on one variable at a time, but many of the coefficients are zero, so it doesn't have to spend time on most of the variables. Handles sparse X naturally and exploits sparsity of solutions, warm starts, variable screening, and includes methods for model selection using cross-validation. So that's a package, and I'm going to illustrate the package on an example on the UK Biobank, and this is the tall and wide data set. So there's a large cohort of 500,000 British adults who've been tracked, who volunteered and been tracked for the last, I think, 10 years, or maybe a bit less. And in particular, they've been genotyped, and we have available genotype data at 805,000 locations. So now you've got a data set of 500,000 subjects and 800,000 variables or SNPs, and the genotype is a 3-category factor variable, each of the genotypes. And the idea is you want to try and build, for a long time, this data set was done with the SNPs or these locations. So each location is a SNP, single nucleotide permutation, polymorphism, sorry. And you want to see if that mutation actually affects a disease or affects some phenotype. And what was typically done for a long time was there was sort of univariate screening. You looked SNP by SNP and saw if there was an association. And in the last few years, we and others have tried to build multivariate models to predict these, to try and model these phenotypes using all of the SNPs. And so, and this team here, which was my student at the time, Yunyang Kian, and two colleagues of mine in biomedical data science, which is my other department these days, Yosuke Tanigawa, Rob Tupshrani is part of the team, and Manny Rivas, who's this guy over here. That's a group in biomedical data science. Okay, so there's hundreds of phenotypes measured on each subject, and so we'll model typically one phenotype at a time as a response, and all 800,000 SNPs as predictors. We looked at a white British subset of 337,000 and illustrate on the phenotypes height, asthma, and myocardial infarction. In fact, I think I left this one out. We divide the data into 60% training, 20% validation, and 20% test. And the package SNPnet is a variant on GlimNet to accommodate these data and some special modifications of GlimNet to be able to work on data of this kind. And our papers just got published in PLOS Genetics on how we do this, and I'll just be reporting on some of the results. So this is a picture that shows a phenotype height and standing height of the individual, and it's a popular one because it turns out the genetic information that controls height is on all the chromosomes. It's really widely distributed. It's a very polygenic trait. And so in order to do a good job predicting height, you need to use many, many SNPs. And what we see here is R squared. We report in percent of variance explained as a function of, this is plotted against lambda index, but this is the regularization parameter as we're going down the Lasso path. At the left, we're using none of the SNPs. And as we move to the right, we're using more and more SNPs. And at the top, you see the annotation of how many SNPs are in the model. So this is going down the regularization path. And what I'm reporting is R squared. So the first curves we'll look at is, first of all, the black curve is the training R squared, as we include more and more SNPs as selected by the Lasso model. So each of these dots corresponds to a Lasso model fit with a different value of the regularization parameter. And, you know, obviously it's just going to keep on improving because this is training R squared, right? And you can see it's getting up over to 85. And if we let it go on forever, it would get up to 100%. But more important is the orange curve, which is the validation R squared. So this is, you know, left out validation set. And we see it starts leveling off, and finally it's leveled off, and it's going to start going down because you'll be overfitting. And that's actually when we stop. And at that point, you can see that there's almost 48,000 SNPs in the model. So that's a massive number of variables. And that achieves an R squared of 70%, which is pretty much a state of the art for this phenotype. What I also show you is the relaxed Lasso, which is an interesting, and this is a new innovation to GlimNet as well, to the package. And what the relaxed Lasso does is it takes the active set selected by Lasso. So for example, at this point over here, there's just over 13,000 SNPs in the model. And then what it does is it just fits that model unrestricted, just on those variables. So it lets the Lasso do the variable selection, and then it fits the model by maximum likelihood, unrestricted. Okay. And what's interesting is that, of course, its R squared does better because the Lasso is always regularized. So it's done a selection and regularization. So on the training data, the R squared for the relaxed Lasso does better, right? But on the test data, on the validation data, it starts overfitting sooner. You can see it's overfit by 13,500 SNPs. But interesting as well is that the maximum R squared achieved is smaller than for the Lasso. Right? So the Lasso, by doing the regularization, is allowed to pull in more of the SNPs that are really needed and controls the variance by regularization. Okay, so this is a little bit about the computation. So how do we actually do this? And the point is that we cannot get the whole SNP matrix into memory. It's massive. Right? None of our computers would be able to accommodate that whole matrix. So what we do is that we go as follows. We basically screen the SNPs, and I'll tell you a bit more about that in a moment. We screen the SNPs, and we pick a set of roughly 1,000 SNPs that are most likely to be in the beginning part of the model. Right? In the beginning part of the model, you're going to get the most important SNPs, right? The ones with the strongest signal, they're going to come into the Lasso model first. So I'll tell you how we do the screening, but we do the screening and we grow the Lasso path. So what I'm showing you here are the coefficients as they evolve as we relax the penalty. And you see the first SNP comes in and its coefficient starts growing, and the others come in. And then we go a certain number of steps, and it's 10 steps. We go 10 steps here. And then we stop, and we look at all the variables in, and we're able to check, right? This looks like a smooth curve, but it's actually in steps of the regularization parameter we used. And we're able to check on all the SNPs whether these are valid solutions for the Lasso model on the whole set. And the blue guys are valid solutions and the red guys aren't. So then we stop at that point where the vertical line is, and then we screen in some more variables, and then we continue the path. And so now the blue continues for a while, and then again we do the check, and these guys didn't come in, so we stop at this vertical line. And so you see we grow the model in these kind of bursts. And the value in that is that we never had to put all the data into memory. So how do we do that? I'll show you. I'm going to tell you the next 2 slides is about screening rules. So let's start with, and this is work I did with a group, Ken Lang and Tong Tong Wu in 2009. This was a small GWAS with 2,000 observations and around 1 million SNPs, right? Much fewer observations than we have here, right? And here was the idea. You compute the inner product between each SNP, that's the xj, and y minus y bar. So y in this case was a 0-1 binary variable case control. And the data set, it was actually 2,000 observations at 1,000 cases, 1,000 controls for some disease, right? So y bar is actually a half, right? And you compute the absolute value of this inner product, right? So this inner product sort of measuring the association between the SNP and this kind of residual for each of the SNPs, and there's 1 million, right? And we order the SNPs according to that absolute inner product, right? If the variables were standardized, this would be equivalent to largest correlation with the phenotype. Find the SNP with the largest correlation and so on. Now, it turns out that if you do that, the largest inner product happens to be what we call lambda max. And that's the smallest value of lambda. Lambda is a regularization parameter in the Lasso. And that's the smallest value of lambda for which all the coefficients are still 0. Right? So that's how you can identify which variable is the first one to come into the Lasso model. What we do is you fit the Lasso logistic regression path using only the largest M of these, say 1,000. And typically you're going to fit with a sample of size 2,000, you'd fit maybe a model with 20 to 30 terms in it. And then you would do a simple KKT check that the emitted SNPs would not have entered the model. So basically the idea is you screen what you make you have a guess at what are going to be the variables of a bigger set that's going to include the variables in the model. You fit the model in that bigger set, and then you can confirm with simple checks that they are actually the only ones in the set. So that's the idea, and then that idea has been developed into what we call strong rules. So if you have a Lasso fit with active set A, script A, which what we mean again is that you fit the Lasso with regularization parameter lambda. Just to be clear here, I'm talking about lambda all the time. Oh, unfortunately, I don't have this. I have this in bound form, so I don't have a lambda. Sorry, these slides got cold a bit. But anyway, I think many of you have worked with Lasso. This is the multiplier on the penalty. The bigger lambda, the sparser your model. Anyway, if you have a Lasso fit that results in a model with a certain number of non-zero variables, defined by the active set by A, it has a property that inner product between each of the variables in the active set A and the residual from the fit is equal to lambda for all variables in the active set and is less than or equal to lambda for all those not in the active set. And typically less. It's only going to be equal if they're just about to come in or just about, in some cases, to drop out, right? So in other words, the requirement for entry into the Lasso model is large inner product with the current residual, okay? So variables nearly in the active set will have inner products with the residuals nearly equal to lambda. So suppose a fit at lambda L is this x beta hat lambda L, and we want to compute the fit at lambda L plus 1, which is less than lambda L. So we're going to drop the penalty parameter, and in doing so, we expect a model with more variables in, right? We expect some extra variables to come in. So what we've actually done by going to lambda L plus 1 is we've lowered the bar for entry, okay? So now what we could do, of course, is we could look amongst all the variables that we haven't considered so far, and we've got a current residual, right, from our current fit, and we can look at the absolute inner products of all the variables out there, and all those whose inner products are bigger than or equal to lambda L plus 1, we're going to certainly include those as candidates, right? That would be natural because we've lowered the bar, so many of them should come in. But actually, we lower the bar twice as much, and so that's this expression over here. And there's some theory behind why that makes sense. And so those are the guys we're going to consider, right? So it's like we just hedge in our bets, and that's a kind of screening. So the LimNet screens at every lambda step, and after convergence checks for any violations, right? And in most cases, these strong rules, this set that we hedge on, the actual active set is a subset of them. So we almost never make mistakes. Right? In this figure over here, we didn't quite use the strong rules. We used the M features we let in was a much smaller set because the strong set would often be way too big. And so we did, and we did make a few mistakes. And we the intention in these rules, in these rules here, is to just fit the model at the next value of lambda. And so what we did over here was we'd let in a set and we'd fit the model, from going from year to year. We start from this point, right? So we started from there, and then we'd fit quite a few values of lambda, and then we'd go back and check and see which ones we made the mistake. But that's the idea. So it's an adaptation of the strong rules which allowed us to fit these models on a much larger scale. Okay. So that was height, but most of the phenotypes on this data set are disease phenotypes such as asthma. And what this lets you do is build what's called a polygenic risk score for particular diseases such as asthma. And so here we've got the observations ordered according to the asthma polygenic risk score. And what we see is indeed, if you break the observations into bins of PRS and look at the odds ratio for asthma, you see it's just monotone increasing, and it's very strong, right, as you might expect. And this is on test data. Okay. And here there's far fewer SNPs than there were for height, but still over 6,000 SNPs, and again they seem to cover all the chromosomes. And so this is a Manhattan plot of the absolute coefficients in the Lasso model, and some of them stand out, but many of them are just non-zero. And but what you see from this is most of the chromosomes are involved. And there's other ways of looking at the data. Here we plotted age for the first event of asthma, and the observations are grouped in tiers of the polygenic risk score. So those in the top 1% of the polygenic risk score, right? They have their asthma event much sooner in life. Top 5%, so the orderings as you'd expect. This is a plot just showing the effects. So here in this first group here is different quantiles of the polygenic risk score, and we see on a test set, they're very significant in terms of association with asthma, and compared to other traditional risk factors for asthma, much stronger, as you might expect. Okay, so more on GlimNet. We're currently version 4.02. There's a website for GlimNet. I encourage you to have a look. So this is some of the new work that we did with Ken Tay. Originally, GlimNet only had certain families like Poisson and binomial and Gaussian, of course. We've now allowed for any of the families you can have in GLM, so programmable families are accommodated in GlimNet. So, for example, you know, if you're used to doing this kind of thing, you can say family equals binomial, link equals probit, or family equals tweedy, and you can give some parameters. And people can program up their own families. And so that can all be done in GlimNet. I mentioned the relax option where you can refit all the models in the path with without regularization. The original GlimNet also included survival models. And we've added a few other bells and whistles according to following requests from some of our users. And coming soon, full functionality for Cox models, such as start, stop data, and strata, and so on. So this is a project that we actually actively working on, and we'll probably keep going with this. Okay, so that's the first topic. The next one I'm going to tell you about is random forests used as an ensemble learner, and this is work with some colleagues in epidemiology, and there they listed Alice Whittemore, just retired, but she's been a colleague at Stanford for many years, and these are some researchers and students from the department. And so this is predicting the pathogenicity of missense variants. So the idea is to prioritize a list of candidate genes for prostate cancer. And, you know, missense variants is if there's mutations in your DNA, these mutations show up in the RNA, and they show up in the triple that makes an amino acid in, you know, when the RNA is turned into a protein. And a mutation in one of the elements of that triple may not change the function of the amino acid, right? There's a many-to-one mapping from these triples to amino acids. But in the case where they do change the function, it's called a missense variant. And these are thought to be responsible for a lot of diseases. And so that's the idea here. And the system we have is called REVEL, Rare Exome Variant Ensemble Learner. So the approach is as follows. There's a number of existing scores for disease status, and they don't always agree, okay? So these scores, what they do is you've got these missense variants, and you look at other aspects of them, you know, what amino acids are nearby, certain physical distances, and so on. And so people have come up with a variety of methods for coming up with scores to score these missense variants for pathogenicity. And in fact, there's 12 that we focus on. And the idea is to use a random forest algorithm to integrate these scores into a single consensus score for predicting disease. So this is known as an ensemble learner. And a random forest is kind of a really non-parametric method for doing that. So we're going to use 12 of these scores as features and run a random forest. Got a big data set. It's a combination of two data sets. And these are the sample size. We got a training set of 123,000 neutral variants and 6,000 disease variants, and then we've got a test set that we put aside, which is a more balanced test set. This is a correlation matrix of the scores. It's a 12 by 12 matrix, and you can see, so red is high and yellow is somewhat high. You can see some of these scores are correlated, right? So they sort of report in the same thing. And some of them aren't, so the ones that are, this is, I guess, is an absolute correlation, but some of them are weakly correlated. So the blues are weakly correlated. So that if everything was really strongly correlated, you wouldn't get much benefit from an ensemble learner. But the fact that there's a lack of correlation means there's some hope. So for those who are not too familiar with decision, with random forests, so the idea is to use a forest of trees to do this ensemble. So here's a single tree, and the single tree would go through these features and make thresholds, right? Is SIFT bigger than 0.9? Yes, you go to the left. No, you go to the right. Keep on making decisions. And when you get to the terminal nodes, you get a probability for disease. Okay? So if you manage to go down this branch, your probability of disease would be high, some branches low, right? So this is a shallow tree, and shallow trees are too coarse or inaccurate. And deep trees, or fine some groups, are more accurate, but very noisy, right? So Leo Breiman, there's a picture of him, invented random forests in the early 2000s. And the idea was to fit many thousands of different and very deep trees and average their predictions to reduce the noise, right? So you want deep trees to make them relatively unbiased, but they're very noisy, high variance, and so average the predictions to get rid of the noise. Now, you need the trees to be different or somewhat uncorrelated to get the benefit of averaging. So how do you get different trees? You grow trees to bootstrap subsampled versions of the data. That will make them a bit different. And you also randomly ignore variables as candidates for splits when growing the trees. And so that breaks some of the correlations between the trees. Very simple idea, and it works, amazingly well, right? So it's so much so that it's become one of the go-to tools in all these prediction competitions like Kaggle and so on. It's always one of the tools people try, or often is anyway. And pretty much automatic, and give good CV estimates of prediction error for free. So the random forest has out of bag, so-called out of bag error, which turns out to be almost identical to leave one out cross validation. And you get it completely for free. It just comes from the way the method is fit. So here's the result of using random forest as an ensemble learner for this task. And I'm showing the results as an ROC curve. Remember, we're trying to classify, it's a two-group classification problem, and we can vary the threshold. And as we vary the threshold, we change the false positive rate and the true positive rate on test data. And what you want is a curve that hugs the northwest corner. And our curve hugs the northwest corner more than the other curves. Now, it's interesting, these other curves are also ensemble learners. They also tackled the same problem. How can we combine these 12 raw scores and come up with an ensemble to do better than all of them? And of course, we've done better than all these other ensemble learners. Otherwise, I wouldn't have shown you this picture. So that was a big success, and this paper got published. And random forest also gives you a variable importance plot, so we can see which of the inputs scores were the more important, and two of them seem to stand out. But all of them play some role. Okay, so that was a success on tall data. Next example, I'm going to give you some examples of using deep learning for image classification. And in this case, these are wide data images. The pixels of an image are the features, and they tend to be many, and that tends to be wide. So this is a nice cartoon of a convolutional neural network used for image modeling. And this is actually a picture that's going into the second edition of our book, Introduction to Statistical Learning, where we have a chapter on deep learning and neural networks. And it's, and this is CNNs, convolutional neural networks, is definitely one of the big contributors to the enormous success of deep learning and neural networks that we see around today. And the idea is as follows. Here's an image of a tiger. You've got the 3 color panes of the image. You have a little filter, which is shown as this little square here, which is you can think of as a small image, but it's got weights in it. And you do a convolution of this filter with the image, which means you do an inner product. You slide that little filter along the image, and you do an inner product as you go. So it's a 2-dimensional convolution. And when you do it, you get a new image, which is this image over here. And it's actually using all 3 of the panes in doing the convolution. So there's some details, and you will be able to read in our chat minutes. You can read anyway. And for each filter you use, you get a different pane. So these panes here are the result of convolution with many different filters. So this filter is intended to be a feature extractor. And then there's a pool layer, which you take this image and you in this case, we've taken each non-overlapping 2x2 block of pixels and produced the max. It's called a max pool. And so you get an image of half the size. And then you repeat. You convolve, pool, you get an image of half the size. You keep on going, and eventually you have a fully connected layer in your fit. So people have used filters for a long time for image modeling. The beauty of CNNs is that these filters are learned in the process. You have a task. In this case, it was a 100-class image classification, and you learn the filters and all the weights in the model as you're going. So major success for image classification. In healthcare, we have many digital images, such as mammograms, MRI scans, X-rays, optical scans, skin photographs. And radiographers and people associated with this kind of work have spent many, many hours poring over these images, looking for small features in that. And nowadays, that job's made much easier. So modern software tools are accessible to all. We used Keras in the R package Keras, which links to the TensorFlow package in R. So I'm going to give you 2 examples. One I didn't have anything to do with, but I was so impressed with the results that I thought I'd just let you know. So this is diabetic retinopathy. So diabetic patients often have this problem with their eyes, and you go into the optometrist and you get a picture like this taken of your eye, and then somebody looks at it and finds some abnormalities. They'll find little hemorrhages and things like that. So this is a perfect task for deep learning, because you've got a standardized image. It fits, it fills up the frame. All images are centered, so they fill up the frame, and you can train a deep learning model to do this task for you. I found 2 papers. Both these people gave talks in our biomedical data science department. Very impressive talks. And this is one of the tasks where the deep learning is actually starting to surpass the performance of human raters. And so here's for one of the papers, here's a comparison of the deep learning to particular experts. Each of the dots, an expert. The ROC curve is for the deep learning model. And we've zoomed in. So it does better than some of the experts. Some of the experts do better than it, right? But the deep learning model does really well. And the nice thing is you can decide what the threshold is. The expert usually is going to say yes. You can see these experts obviously have different thresholds because they gather around at different points along the ROC curve. But with a deep learning model, you get a functional model, you can set the threshold. Okay? And eventually you can train deep learning models on a consensus of experts and get better performance. So this is a problem where machine learning has really, and deep learning has really paid off big time. This is a project in which I was involved. This is image segmentation. So these are MRI scans of athletes that have had injuries, whiplash injury. And these are cross sections of the spine, and the green and blue regions that are colored in here are what's known as deep cervical spine extensors. And the idea is to measure the fat infiltration in these sensors after whiplash injury. And that's what happens when you have whiplash, you get more fat infiltration in these sensors. So the task of the radiologist is to first outline these regions, identify these regions, and then somehow measure the amount of fat in those regions. These regions here have been, these are all the same MRI scans. These rows are different raters, and at the top is a convolutional neural network. So here the network is not trying to classify the whole image, it's trying to classify every pixel in the image as being in the right sensor or in the left sensor or not in the sensor. So it's a pixel-wise classification, okay? And it does really well. So there's some small sample issues here. We didn't have a lot of training samples here, just a few hundred. And so some of the tricks that you use is, you adapt a pre-trained network. So already there's lots of networks being trained on MRI images of the spine and other parts of the body. And the idea is you use some of the early weights in the images. If we go back to our diagram, some of these early filters that are learned for natural images in this case are going to be general filters. They're going to work with any natural image. So if you're short on data, you don't have to retrain these, or you can get by with not retraining these weights and only retrain some of the weights closer to the target, okay, closer to the loss function. And so that's a standard trick now used with deep learning. So you adapt pre-trained networks. And the other one is image augmentation. Image augmentation says you've got one training image, let's just say one of these guys, and you do some natural deformations to that image that aren't going to change the way a radiologist would do the annotation, right? So you can change the contrast, you can do a little bit of warping of the shape, and so on. So you can do some natural changes that are particularly adapted to the type of images you've seen, and thereby enlarge your training set. And it turns out you can view that as a kind of regularization. Anyway, so this work was published recently, and I'm just showing you here there were two things. There's a volume of the region, and there's a muscle fat infiltration. And what we're seeing here is correlation between the deep network and the raters, and you can see that that's pretty good. So that was one of the performance measures. So in these kinds of tasks, deep learning is really effective, and there's endless applications now where it's used. I'll just finish off. I mean, it's very easy to get extremely enthusiastic about deep learning because of the successes. But it's not always the tool to use. So this is an article that appeared some years ago in Nature Medicine, 2001. It was a 4-class cancer classification task. 63 training observations, 25 test observations. The features were gene expression on 6,500 genes. So very wide data set, very few training observations, right? And it's four classes. So these 63 are broken into 4 classes. So that's nominally 15 observations per class, but actually it was even more lopsided than that. But in this paper, they nevertheless fit a neural network. And this is some of the diagrams that appeared in the paper. And, there's a network diagram for a neural network. They used bagging, which is one of the tools used in random forest to get rid of some of the variance. It was a complex method. And the paper was headlined in the journal, model for the human brain, solves difficult cancer classification problem. And indeed, they made 0 training and 0 test errors, right, on their 25 test observations. Well, it turns out you could do a simple linear model. You could do a regularized linear model and get the same performance results. You could compute the first 12 principal components of the genes and fit a linear model on them, and you get the same test results. In our book, Elements of Statistical Learning, we touch on this data set, and we show an even simpler model that also gets 0 training and 0 test errors. So this was a case where, honestly, the neural network was not doing us a service. Right? When you've got such small amount of data, it's very likely that a much simpler model is going to do the job. And I would say in a case like that, you want to go with a simpler model. So, I think Leo Breimer, I've heard this expression many times, but when you have a big hammer, everything looks like a nail. I think, we have to be responsible. It often seems sexy to use the latest technique. You write a paper with deep learning in it, in the title or in the abstract. It seems like you're using state-of-the-art techniques. But if you could have used a much simpler model, such as a linear model, and got equivalent results, I think it's irresponsible to go down the exotic route. Okay. So to summarize, we saw examples of Lasso, random forest, and deep learning. And we also saw an example where neural network was overkill and a simple model was sufficient. So modern machine learning tools should blend with existing methods and keeping in mind Occam's razor. So that's all I had to say today, and I'll end up with some cheap marketing. These are some of our recent books. The only reason I feel entitled to advertise them like this is that all of these books are available free on my and my co-author's website with arrangements with the publishers, so the PDF's available for free. And with that, I'll thank you very much for your attention, and happy to take questions.
Great. Well, thank you, Trevor. It was an excellent talk. Normally at this point, you'd hear the applause of the audience, but you'll also have to take my... That would have happened if we'd been in person. I have collected a few questions through the chat, and there's some that are very specific and others that are more general. Maybe I'll just start with those. So Jake Cooper asks, are the advancements being made to the R-GlimNet package also going to be added to the Python version?
Yeah, that's a good question. At some point we were advancing the Python version, but honestly, it becomes difficult because a lot of the methods that we use for the R-GlimNet package are going to be, would have to be somehow redeveloped and reinvented for the Python. So we actually slipping behind on the Python version. I've been collaborating with a team who likes to use Python, and what they do is they call GlimNet from Python. And so I think that may be the way to go.
Okay.
Yeah.
Other questions here. We have Rajan Patel asking,
sorry, I should have, so just before I leave that point, I didn't say it quite right. The idea is to call the R package GlimNet from within Python, which you can do.
Yes.
Yeah, sorry.
You talked about, tall and wide data and so on. Are there any thresholds that you can use to decide whether your data set is big or tall?
Oh, no, that's just a loose classification, but it sort of guides you to what types of methods you're going to be using. So generally tall is, there's no thresholds. And but that's just a rough rule of thumb.
And in your second example, you didn't have enough time to go into all the details, but there's just a question about why, is there an intuitive reason or why the Revel method worked better than the other ensemble learners?
The others, not really. We just use random forest was really used as a black box in that case. So it's possible that the other methods were using less flexible learning methods.
Okay. And then maybe going to something a bit more general, I know that you have made quite a few contributions to software, as you mentioned in your talk. And there was a comment in the chat about, earlier on you said it's hard to get published. You do a lot of work on software. You don't get much appreciation necessarily, at least in terms of publications. And Patrick Brown, who are in the chat, you can write a paper on the package and submit to the Journal of Statistical Software. Mm-hmm. What is your experience been trying to get papers like that published?
Oh, no, not too bad. I mean, when we first wrote the GlimNet package, we did send a paper to Statistical Software. But when you add some features to the package, mm-hmm. It may be hard to get a paper out of that.
Right.
The features that you add are really valuable, such as the stuff Kentay's been doing. But maybe difficult to, the features you add in are they're already known in other contexts, like mm-hmm. survival models with all different kinds of ways, having stop-start data and various things that people like to do today. So there's no novelty really in that. The novelty is having that inside this package and, and having all the other facilities of GlimNet, on response. It may be difficult to get a paper. Maybe we'll try, It depends on the Journal of Statistical Software and what they consider a novelty.
Right. And I had just one general question for myself. Early on, you mentioned, and I agree with you, the importance of sort of internal validation. You said that a lot of times it's not done or not done correctly. Is it not done at all, or is it just not done correctly? What have you seen?
I think it's not done correctly, and it's very easy. If you've got a data set that's really important to you and you've got a vested interest, you spend a lot of time with that data set, it's very easy to cheat, and unintentionally cheat, But right. Even more blatant cheating is, you've got thousands of variables. So early on, you do some kind of screening of the variables because they don't seem to be important. And if you use the response to do the screening, and then now you've got a reduced data set, and now you pretend that's all you have, and you run cross-validation, that's biased. And so that screening, for example, has to be included in the cross-validation. And many, many times we've seen that not done correctly. Yeah.
Okay, lots of thanks for your talk, by the way, and the question and answer as well. I just passed that along in general terms. Let's see what else we got. How heritable should a trait be for an optimal use of SNPnet?
Well, it doesn't have to be that heritable at all. We've actually fit now models on all the phenotypes in the UK Biobank. And some of them are not that heritable at all. We get R-squareds are quite low, 5-10%. So it'll always fit a model, right? And it just depends on how heritable the SNP is.
Okay.
I should add, when we fit these models, what's standard in this kind of world is, you'll adjust for variables like sex and usually the first 10 or so principal components of all the SNPs, to allow for geographic variation and things like that. So there's a standard practice. So usually sex and age are put in as adjuster covariates first, unpenalized.
And a related question to the internal validation. In your example, you split the data, 60-20-20 for training, validation, and test. And the sort of traditional maybe is to use 80 for test, training, and 20 for test. What's the question?
Yeah, that was something, honestly, I wasn't that excited by that at the end, but we decided not to redo all the work. These are massive data sets. You don't need such a big test set and right. A validation set. So we could easily have had, smaller test and validation. We did need to separate the validation and the test set because we were using the validation set. Instead of cross-validation, which would have been very costly, we put aside a validation set to tell us when to stop training the model. And then you want to go and use fresh test data to evaluate the performance of your chosen model. But they didn't need to be 20%. Yeah, I agree.
So another interesting question here relates to sorry, just trying to find it again. What do your clinical collaborators think about the trade-off between model complexity and interpretability? You know.
Yeah, so in these kinds of models, you know, the interpretability is not really the goal, right? The polygenic risk score, you're just trying to get a good score, right? With all those thousands of SNPs involved in the model, it's unlikely to get an interpretable model. Even in smaller data sets, when you use Lasso, you know, people have tried, and there's some progress in doing post-selection inference, getting p-values for chosen variables and so on, but it's difficult, you know, and it's difficult to do, and you know, and it's problematic, you know. It's just like whenever you've got correlated data, trying to attribute, you know, responsibility to different variables is difficult.
Yeah, and another sort of related question, in the PRS example, if prediction is the goal, oh geez, my chat window keeps shifting and then I can't read it anymore, sorry. To find the question again, if prediction is the goal, why not use the ridge penalty, particularly since the cost of collection of 8,000 SNPs is the same as 800,000 SNPs?
Oh, yeah, that's a good point. We have tried the ridge, and ridge, it doesn't do as well, because you're gonna end up having a lot of useless SNPs in the model. They're going to be noise, and in order to kill that noise, you're going to kill a lot of the signal on the good SNPs. So in this case, Lasso did much better than Ridge. I didn't show the Ridge yet. Ridge is would also be problematic because, you know, you wouldn't be able to do the screening because by definition the Ridge fit has got all the SNPs in the model. But there's ways of fitting the ridge via principal components. You don't need to use all the principal components, and you can get by. But we found that this did much better.
Another comment here: coordinate descent seems to work remarkably well for feature selection with DLS. Can this be generalized to other types of models? Yeah,
it's well, any linear model with a Lasso penalty, coordinate descent will work well. And the reason is that even though you might have a lot of variables, the solution is generally going to have a relatively small subset that are non-zero. And when you do coordinate descent, you can focus on the active set and get convergence on the active set, and then just check the solution at the end. So it's actually both active set convergence as well as having a path of solutions. So when you fit the path of solutions, you've got really good warm starts from one value of the penalty lambda when you go down to the next smallest value of lambda. Mm-hmm. The active set usually grows by a small fraction, and you get really good warm starts. And so that's all suitable for coordinate descent. It suits coordinate descent very well.
So one of the things about it was this, I can't remember what it's the Lasso.
Relax, yeah.
What's the advantage of that? It did seem to have worse validation performance.
Well, it doesn't always. In this example, it did, right? But there's other examples where it does better. And so the advantage of that is that you can, you know that when P, the number of variables, is very large, for example, best subset becomes really hard to do, right? Mm-hmm. It becomes combinatorially hard, even though there's some progress these days with mixed integer programming and so on. It's still a very hard problem. Whereas Lasso just dives in and finds its active set. It's a convex program. But it does penalize. And so the idea is, you can get Lasso to find this path of active sets, and then you can relax the fit on them and get an unrestricted fit. And often it finds the same set of variables that best subset will find. So not always, obviously, but it often will. So it's like a good hedge for doing best subset, but efficiently.
Okay. This is maybe a big topic, but do you have any general suggestions for guidelines or things to think about for the internal validation?
Yeah, just let me finish off on that other point.
Okay,
We have a paper that's just come out in Statistical Science, and there's some discussions of it as well, which compares best subset, Lasso, and forward stepwise. And the paper with Rob Tibshirani and Ryan Tibshirani and myself. And so people might be interested in that. So I beg your pardon. So the next question was to do with internal.
So general suggestions for how to go about and what to think about when you're trying to do internal validation properly.
Well, yeah. So we advocate and use cross-validation a lot. So where you say break the data up into 10 parts and use 9/10 as training and 1/10 as test, and you do it for all combinations. And that's a sort of satisfying approach, and there's a lot of variants of cross-validation. So that's one way to use your sort of valuable training set and not sacrifice a lot of it for test data. Mm-hmm. And figuring out how to do that and to do it in a correct way it's often not trivial. So, for example, if you have observations that are correlated, maybe members of the same family or things like that, you have to do cross-validation intelligently and leave out whole families together and things like that. But I think it's something it's intuitively somehow doing the right thing. And we favor that more than, for example, we favor these model-based procedures such as AIC and CP and methods like that.
When you have 400 people on your talk, there are typically lots of questions we're going through. We'll have to cut up at some point, but I got a few more here maybe. Do you think that reinforcement learning can improve performance better than the deep learning by combining simulations and neural nets, specifically in genetic applications where features are highly correlated?
Honestly, I don't have much experience with that. I mean, I know about reinforcement learning. I've seen it used in things like game playing and like Go and chess and things like that. I'm not. I haven't seen it used in applications like the ones that I've talked about here.
Okay. In your UK biobank example, you subsetted the data and used only the white British people, mm-hmm. And you trained your model for that data. Do you think the models are transferable to the other populations?
To other white male populations, yeah. I mean, we did that because there's a real there's definitely a big race. White males were a well-represented group in that data set, and I think it was it white males or was it just white?
I think it was just white British. We're British.
Yeah, and we adjusted for sex. And we know ethnicity plays a big role in height, right? It is a feature that's responsible. And some of the other ethnic groups were less well represented, so we just decided to focus on that subset. Yeah.
Okay, I think maybe we could, we're kind of over time now and we have another session starting right after this. For those of you who signed up for the private Q&A, I suggest that we thank Trevor for a really interesting talk again, and look forward to having you visit Waterloo sometime in the future.
Oh, yeah, well, let me just say it again, it's a real honor. I've been asked to give the David Sprott lecture, and yes, I am sorry that I wasn't able to visit Waterloo in person. We'll have to save that for another time, but thank you all very much for coming to the talk today.
Thank you very much, and sorry, anytime you can come to Waterloo, let us know. So I guess we'll finish the talk now, and those of you who have the link for the other session, we start right after this. Okay, thank you.
Thank you.